current speaker is a full-fledged professor of the biology department, Cebu Normal University, Cebu City. His PhD dissertation was on systematics, ecology, and conservation status of Philippine cinnamon. Aside from pursuing basic research on cinnamon's medicinal property, he also actively provided technical advice related to Philippine cinnamon taxonomy and how could this knowledge be utilized to propel community enterprise and conservation initiatives of the genus. And he also aspires to explore all potential sources of taxonomic data and hopefully resolve many taxonomic controversy in the genus Cinnamomum. He is currently involved in Cebu City Urban Biodiversity Plan and is actively involved in pursuing community projects leading to the sustainable propagation of native species in urban areas. So again, let us all welcome Dr. J.P. Picardal.
Good afternoon. Today I wish to share my journey to everyone who is attending this plant biology symposium, especially to aspiring botanists and plant taxonomists who are looking for major reasons why plant taxonomy is a promising field of biology. My talk centers on the recent taxonomic initiatives in support to community-based cinnamon enterprise in the Visayas region. I decided to choose this topic because it supports the theme of the symposium, plants in times of crisis. How plants and studying plants can save humanity during difficult times. After this presentation, I hope that we will realize the importance of the science-based community enterprise in supporting conservation initiatives of native plants in our country. In today's presentation, I will start presenting some taxonomic controversies of Philippine cinnamon that has long puzzled taxonomists and parataxonomists alike. It will be followed by a presentation of research activities which aimed to address the previously mentioned controversies. Then I'll be showing as well some photo documentations of our fieldwork and how we were inspired by community-based cinnamon conservation initiatives in Central Visayas. The finale of my presentation would be a testimonial from Ma'am November Caniesoyo, who will present how plant taxonomy helped their company tap the potentials of different cinnamon species in Western Visayas. Cinnamon is a close relative of avocado and bay leaf from the family Lauraceae. And so far, there are more or less 23 species reported in the Philippines, 16 of which are known to be endemic. Aside from its distinctly fragrant, woody, fruity smell, most cinnamon species have triple veins, we call them triplinerve or trinerved, possess paniculate inflorescences, and fruit seated in a cupule like a small avocado fruit. Cinnamon taxonomy found a strong support in both vegetative and reproductive characters, with leaf variation often the most challenging to work due to indistinguishable phenotypes. In the work of Wu Kwang in 2011 for Bornean cinnamon, he showed the variations of the leaf characters, particularly the divination, different inflorescences type, as well as the different fruit categories in terms of shape and the shape of the cupula or the fruit cap. In cinnamon, particularly the cinnamon species in the Philippines, the most common concern is that there's a wide range of morphological variation of foliage at the species level. Another concern is that the smell and tastes are inconsistent as reported by farmers. And they thought that these are largely influenced by edaphic factors as well as some climatic conditions. Since it is a spice and cinnamon is known to be drunk for particular medicinal purposes as well as used for flavoring, particularly in cooking local cuisines, we cannot help but allow or consider the judgment of farmers when they say that this kind of cinnamon species is this kind of variety or this kind of species for that matter. So they often use their taste and smell to recall the local names in the dialect. Another concern is that intermediate forms are common in saplings. That's why we avoid using material or specimen from saplings, and we get usually from mature trees. And finally, another concern is that the chemical characters identified from different Philippine cinnamon species are inconsistent even among similar species. So, one of the most common taxonomic problem on cinnamon in general is the intra-specific phenotypic variation in which heterophyly and heteroblasty should be taken into consideration. So what is heterophyly? Heterophyly refers to a condition in which the different leaf forms are found in the same plant. For example, in the case of cinnamon, some species has like 5 to 6 leaf forms or leaf shapes found in the same tree. Meanwhile, heteroblasty refers to the development of distinct type of juvenile and adult foliage. This is very common in trees that are receiving less sunlight, and based on my experience, some leaves are in the shoot. For example, the shoot portion in the tip of the branch, they have different leaf shapes compared to those leaves that are found in the lower canopy part of this like cinnamon stand. Hence, careful decisions must be ensured in sampling for the source of leaves to study. For instance, it is recommended to get sample leaves from the top, middle, and bottom part of the entire tree crown. In these type specimens, previous and current taxonomists named them differently, as evidenced by the paper labels bearing the recommended specific epithet. For example, the botanist and year, and um, who named like Cinnamon burmannii to be Mindanao and vice versa. So you can see the historical background behind the naming of this particular specimen. Another thing is that, like in the case of Cinnamomum mercadoi, it was later on named by Kostermans himself to be Cinnamomum utile. This is just an example of this scenario in which really the complexity of identification, even in the presence of reproductive structures like flowers, still confuses taxonomists. Another example is in the case of Kaningagliitan. They call this Kaningagliitan because of the very, very small leaves. And there are two species actually of cinnamon in the Philippines that have very small leaves. It includes Rupestre and Nanophyllum. So, by the look of their leaf shape and size, aren't they of the same species? This is probably the question that would enter our mind. Could C. rupestre be classified as C. Nanophyllum and vice versa, or they are entirely different species? In the case of this slide, for instance, you can see that both of these samples are Cinnamon mercadoi, but if you look at this left illustration, the leaf shape is really irregular. And as a botanist or as a student would probably ask yourself, so which among these leaf shape should I consider to be able to infer that indeed this is Cinnamon mercadoi? Because compared to the specimen on the right, the leaf shapes are more or less similar to each other. Okay, so that's the reason why, as part of the recommendation in my paper, we really recommend to have more sample specimens to look into to be able to verify in terms of the presence of the most frequently occurring morphological structure like leaf shape before we can finally judge that this particular character state would really be like lanceolate or something like that. So looking at this two species as well, Cinnamon mendozae and Cinnamon cebuense, you can say that probably some of the leaf shape in mendozae is really similar to that of Cinnamon cebuense. But if we're going to look closely, um Cinnamon cebuense is more or less consistent in terms of its leaf shape and the length of the acumen, for instance, compared to the mendozae, which is relatively inconsistent in terms of leaf shape and then the length of the acumen. So in my observations, I noted distinguishing characteristics for each one of them, and I was able to decipher what really is Cinnamon cebuense compared to Mendoza and vice versa. Okay. Actually, this dilemma on cinnamon are not new to cinnamon taxonomy. I mean, even abroad or in the Philippines. In fact, in the paper of Santos in 1930, he was already um, telling the scientific community that he observed intensive variation or wide array or extent of variation within the same species. Okay, these irregularities in leaf shape, for example, was something that made him interested to look into the extent of variation, like in the case of Cinnamomum zelanicum, Cinnamomum mendenense, Cinnamomum mercadoi, and Cinnamomum iners. So the question now would be, as a current day botanist, or if you are interested in Cinnamomum taxonomy, how should we address leaf allometry and phenotypic plasticity if we are in the field already? To address this concern, biologists do not just rely on morphological characters, they also consider other sources of data, for example, chemosystematics, or the use of chemical constituents across cinnamon species. In the case of Philippine cinnamon, for example, you would notice in the following columns, like from Cebuense to Trichophilum, you can say that beta-sitosterol, this one highlighted in pink, is one of the few commonly found compound across these different species. So this is indicative of the unique variation of the phytochemicals that is found in each cinnamon species. And take note that the one examined by the group of Dr. Orgasa and colleagues from De La Salle are just representative species. I mean, they do not really reflect the extent of the variability within the same species. Rather, this is an inter-specific comparison. So, part of my thesis was really to address the most important questions because, based on the premise that I presented, I was able to ask these questions. What are really the essential morphological characters and molecular markers to be used for taxonomic studies? And second, how can we further increase accuracy in the identification of Philippine cinnamon? So, the questions I raised and the answers I sought were found when I started working on my PhD dissertation, and I would like to acknowledge the esteemed Filipino scientists and biologists to help me understand this genus. So basically, I work on the systematics, ecology, and conservation status of Philippine cinnamon. Aside from understanding systematics of this genus, based on morphological and molecular data, I also work on species distribution modeling, employing maximum entropy to study its ecology. And the findings allowed me to propose conservation status of the different species under this genus. Before I proceed, please also allow me to briefly acknowledge the group of organizations who helped me along the way. We have the Flora and Fauna International, the USD, FPRDI, especially to Sir R.C. Ella and Ma'am Flor Samiano, CTU, CNU, BUFI, Global Trees Campaign, and most importantly, the Center for Conservation Innovations. I'll forever be grateful to the Pinoy Cinnamon Conservation Project for the support and for allowing me to venture into this research. This Pinoy Cinnamon Conservation Project in general aspires a long-term target of Philippine forest conservation and recovery through delivery of sustainable development's triple bottom line. So if you would notice, even in the early 2015, some groups of organization and research, related agencies attempted to start looking into the potential biodiversity conservation benefits, social benefits, and economic benefits of this kind of endeavor. So I sought to start the taxonomic journey. I employed the most important piece of document that would allow me to get a background into the Philippine cinnamon, the monograph of Dr. Andre Joseph Kostermans, who's an Indonesian botanist, by the way, and was published in 1986, was the most useful piece of information that I started with to be able to understand this particular genus. And with the help of the monograph and related primary and secondary literatures published here and abroad, I was able to identify 65 morphological characters and their corresponding character states, um, particularly for twigs, stems, petal, and shoot, leaves, fruits, and flowers. There. So these 65 characters were utilized for me to be able to really map or tabulate the commonalities and the frequently occurring character states per purportedly identified species since time immemorial. For those that are found in culture specimens. So the major outputs of my dissertation was able to um, produce taxonomic treatment of Philippine cinnamon, and then was also able to create a dichotomous key for the Philippine species. This is not yet published, by the way. Spot characters for leaves, and taxonomic revision of Philippine cinnamon. So, yes, I wish to publish this with these documents this year, hopefully. I just waited for the right time because, based on my experience, I need to really get a personal experience in looking into the live specimens, or freshly collected specimens, not just those specimens kept for like 5 decades or 6 decades or more, because it really helped me to really look closely into the morphological characters. So when I went home to Cebu after my studies in Manila, I asked my students to address the synonymy concern of Philippine endemic cinnamon, like the cinnamon mindanense, in comparison to Indonesian cinnamon, which is cinnamon burmanii. We started by working on the cinnamon mindanense first because we want to establish the methodology that could later on be used. Okay, just in case we have the Indonesian cinnamon samples. The students' work was inspired by the previous publications of Dr. Buot and his students previously worked on leaf architecture research. The paper aimed to assess leaf morphological data, to develop a general template for the species, which could be used in the creation of useful spot characters and identify intra-specific similarities and differences in the species. So by employing, briefly, these are the methods they employ, and slightly modify some standard methods on leaf clearing, and then followed by staining and imaging procedures, and then afterwards they were able to produce these photographs, okay, and they were able also to come up with this arbitrary decision. Like, say, for example, on the left, you can see how we identify the apex and base regions based on these divisions. On the right, you can see how we establish the upper, middle, and lower lamina of the leaf. So these data are essential in evaluating leaf shape quantitatively. So you would notice, for example, that we were careful in looking into the what they call this one, the quantitative measurements of each division in the leaf, so that we can decide whether the leaf shape belongs to elliptic, oblong, and so on and so forth. My students also employed both continuous and discrete characters with the corresponding character states, like here. No, I can see in this column 1 to 6, to understand intraspecific similarity and differences. And finally arrive at this conclusion that from the samples collected, the continuous characters were more variable compared to the discrete characters, which suggests that the continuous characters exhibit a higher degree of phenotypic plasticity. The group also posited that microhabitat disparities in slope, availability of soil moisture, topography and insulation likely play an important role in some inherent variation, which contributed to the variation in the quantitative characters. I mean, continuous characters. Meanwhile, primary, secondary, tertiary, up to pentarivine categories, arylation, margin, presence of petiole, venation pattern, and medium structures were relatively similar across samples. So somehow cinnamon dense is quite stable in terms of all of these characteristics. So we were after at the end of the study, we were able to say that yes, indeed, the presence of the vein that crosses diagonally from the midrib in Cinnamon mindanense is indeed one of the most essential diagnostic marker for that particular species. Okay, the succeeding slide, I'm going to present some of the photographs that these are evidence of our field work and field visit to be able to visit communities who are planting cinnamon in Cebu. So these are the photos. In these photos are my collaborators. Actually, I was asked. I was hired as a research assistant for this project, and I learned a lot from these people, from these researchers and biologists and foresters. And then these photos were taken from the mountains and hills of Mount Bulak in Dalaget, Cebu. So it's quite an uphill climb here. And here we were actually resting. We're moving from the top of the mountain down to the town. And then this photo was taken in 2015. And we were discussing with the cinnamon Mindanense farmers how they grow cinnamon, how they protect cinnamon from fungal diseases, and how the community, the people's organization, would be able to maximize the potential income coming from this particular enterprise. In this particular photos, you can see. Yours truly, who is puzzled and perplexed with this freshly collected specimen, because honestly, this is really a difficult task, to see this kind of specimen compared to those that are tried and preserved in the herbaria. In this case, I was really trying hard to recall the morphological characters, so I would be able to understand and identify this particular species. You can also see here, these are cultivated cinnamon in Bolhoon, and this is a cinnamon farmer, and the group really visited and tried to understand the best practices, the best cultivation practices that this farmer is doing, so he would be able to cultivate seedlings coming from to cultivate and grow wildlings, okay, coming from his own cinnamon plantation in his backyard. And in these photos, taken from different field visit, I was invited to discuss to the People's Organization of Barangay San Antonio, Bolhoon, how to distinguish their own cinnamon species in the locality compared to other species from the rest of the Philippines. This is actually from the project of Ma'am Flor Samiano from USAF PRDI, and then she invited me to discuss and then show to this people's organization the actual morphological variation among different cinnamon species. Thank you very much for your kind attention, and have a good day. God bless.